Hello, today I will be talking about the steps in muscle contraction. First, I will start with the excitation-contraction coupling, which are the events that link action potentials on the sarcolemma to activation of myofilaments, which is in preparation of the muscle to contract. First, there will be a wave of action potentials that spread from the motor end plate in all directions. When this wave reaches the T-tubules, it continues down into the cell. These action potentials open up voltage-gated ions in the T-tubules, which are linked to calcium channels in the sarcoplasmic reticulum. The channels in the sarcoplasmic reticulum open as well, and the calcium diffuses out into the cytosol. Then the calcium binds to the troponin on the thin filaments. The troponin-tropomyosin complex changes shape and exposes active sites on the actin filaments. Now they are available for binding to myosin heads. Now we have muscle contraction, which is when muscle fiber develops tension and it may shorten. The mechanism of contraction is called the sliding filament theory. Myosin heads need ATP molecule attached to initiate contraction. Myosin ATPase, which is in the head, hydrolyzes this ATP into ADP and phosphate. This energy activates the head, which exerts it into an extended position. And the head temporarily keeps the ADP and phosphate. The extended myosin binds to an open active site on the thin filament, forming a cross bridge between the myosin and the actin. The myosin then releases the ADP and phosphate and flexes into a bent low energy position, tugging the thin filament along with it, which is known as the power stroke. The head remains bound to the actin until it gets new ATP. The binding of ATP to myosin destabilizes the myosin-actin bond. The myosin can now repeat the process. It will hydrolyze a new ATP and then recock, which is known as a recovery stroke, attach to a new active site farther down and produce another power stroke.